You probably know that tons of plastic trash are polluting the world's oceans. The problem is, unlike other garbage, plastic can take an average of 450 years to break down. This means a plastic bottle you throw away today will be around for your great-great-great-great-grandchildren's high school graduation. Most of the plastics in the oceans, though, are tiny, no larger than 6/100ths of a centimeter. These are known as microplastics. But these microplastics are not floating solo. On these pieces of plastic are entire ecosystems of microbes called the Plasti-sphere. But we'll get there in a minute. Starting in 2010, biologists at the Marine Biological Lab, MBM, noticed these microbes and were perplexed. Marine microbes often do cling to floating objects in the ocean, but never plastic. Why did these microbes evolve to do this? In order to answer this question, they needed to know what specific types of microbes were present. Sounds simple enough, right? Well, collecting precise data on microecosystems can be a difficult task. To collect precise data, biologist Jessica Mark Welch and physicist Alex Velm developed a new way to know exactly how many and what phyla microbes were on a plastic sample. The method was called CLASI fish. And no, that's not a fish that indulges in the finer things of life. Let's break it down. Fish, or fluorescent in situ hybridization, was a method that already existed in microbiology. In this method, scientists make a hypothesis on what microbes may be in a sample. Then, based on the hypothesis, they synthesize an Oakley nucleotide, a mini strand of DNA that complements DNA pairs within that microbe. The sample is then chemically treated so that the DNA can pass through the cell membrane and attach themselves to the nucleus. The synthesized Oakley nucleotide, however, has one key difference. This nucleotide can glow in the dark. So once the new DNA is added to the sample, it looks like this. So now we can clearly see what types of microbes are present and where they congregate. However, in this method, only one microbe can be tested at a time. With such a large ecosystem on the Plasti-sphere, MBL needed a way to test all the microbes at once. So Welch and Velm developed a new additional method called CLASI, combinatorial labeling and spectral imaging, which allowed them to add different Oakley nucleotides with distinct fluorescent glows, moving microbe mapping from this to this. A beautiful rainbow miasma of exact data. So now it's possible to see every color-coded phylum on the microplastics and study their behaviors. Now let's go back to the Plasti-sphere. By studying these microorganisms, scientists discovered why these organisms opted to essentially hitchhike on these plastics. Since the plastic breaks down slower, the microbes can travel way farther and way faster than before. A microbe colony now can essentially travel from Chile all the way to the coast of Australia just by hitching a ride. But why does this even matter? Well, you see, the Plasti-sphere may have actually given us the key to plastic ocean pollution. While studying these organisms, the MBL discovered that some of the microbes were breaking down the plastic and speeding up how fast the plastic deteriorated. By harnessing this breakthrough, MBL hopes to develop an environmentally friendly microorganism that can consume plastic with no negative impact on the surrounding environments. And overall, I think they have a pretty classy idea.